Hi everyone, this explainer examines a quantitative analysis of alpha-synuclein aggregation in post-mortem human brain tissue. We'll be exploring an advanced imaging platform that was developed to generate large-scale maps of these protein aggregates, all the way from the microscale down to the nanoscale. This provides some new perspectives on the cellular mechanisms of Parkinson's disease.
So we'll start with the central problem in Parkinson's research, which is the technical challenge of actually seeing the smallest pathogenic protein aggregates. From there, we'll get into the details of the new methodology designed to overcome this. Then we'll review the main findings it revealed in human brain tissue, and we'll wrap up by discussing what these findings mean for future research.
All right, so let's begin with the core challenge in Parkinson's disease research. It really comes down to understanding the specific role of these small, soluble protein assemblies called oligomers. The idea is that they could be the main drivers of neurodegeneration early in the disease, but directly observing them has been, well, basically impossible until now.
You see, in Parkinson's disease, single alpha-synuclein proteins begin to self-assemble into these large, insoluble structures. But there are intermediary species, these oligomers, which are thought to be a critical step in that pathway. They sort of bridge the gap between individual protein monomers and the large fibrillar aggregates we know as Lewy bodies. Now, the hypothesis that these smaller oligomers are the primary neurotoxic species has been tough to prove, mainly because of a technical inability to directly see these nanoscale assemblies inside human brain tissue.
Detecting these small, endogenous aggregates has really been a significant technical barrier. While we've had methods like proximity ligation assays that could confirm they were there through signal amplification, that's a form of indirect detection. It doesn't let you see the aggregates themselves. And this fundamental limitation has really prevented an accurate understanding of their size distribution and how they're organized spatially within the brain. So the fundamental question this research really sought to address was this: how can we develop a method that's capable of generating large-scale quantitative maps of alpha-synuclein aggregates? Maps that span all the way from nanoscale oligomers to microscale Lewy bodies, and do it directly within the complex environment of human brain tissue.
And the solution that's presented is an optical detection and analysis platform that they named Advanced Sensing of Aggregates Parkinson's Disease, or ASAPD for short. This platform was specifically engineered to get around those previous technical hurdles and to allow for the direct quantification of aggregate density, size, and spatial distribution in fluorescently labeled human brain tissue.
The workflow for ASAPD is a 5-stage sequence. It kicks off with 2 experimental steps that are designed to maximize the signal-to-noise ratio. That's background suppression and enhanced imaging. These are then followed by 3 computational steps for automated image processing and data extraction: feature detection, analytical computation, and finally, spatial distribution analysis.
One of the primary obstacles in high-sensitivity tissue imaging is something called autofluorescence, where the tissue itself emits background light. This creates a high noise floor, and that noise can easily obscure the very faint signals coming from small oligomeric species. To solve this, the ASAPD platform uses Sudan black B, a chemical agent that effectively quenches or dampens this background fluorescence. And the application of this method turned out to be highly effective. Just a 10-minute incubation with Sudan black B reduced that background autofluorescence by 93%. To put that in numbers, it lowered the median detected background photon counts from around 4,400 down to 333, which substantially improved the visibility of these small, fluorescently tagged protein aggregates.
So, in addition to cutting down the background noise, the platform also works to enhance the desired signal. This is done by using a microscope objective with a high numerical aperture, or NA. A higher NA lets the objective collect light from a much wider angle, which means it captures more photons from the sample and at the same time improves the image resolution.
For this study, a 1.49 NA oil immersion objective was used. That's a tool more typically seen in single-molecule fluorescence experiments. So by combining that efficient background suppression with the superior light-collecting power of this high NA objective, the platform achieves a significantly higher signal-to-noise ratio. And this improvement is what makes it possible to finally detect those previously invisible nanoscale objects that are just missed by the lower NA objectives used in standard clinical slide scanners.
Okay, so once these high-quality images are acquired, they get processed through a custom-built open-source computational pipeline. The software automates the detection, classification, and quantification of all the fluorescent species, which enables a rapid and reproducible analysis of thousands of images.
The pipeline applies different computational strategies depending on the feature size. So for the large microscale aggregates, it segments them using a direct intensity threshold. But for the smaller, dimmer, diffraction-limited puncta, it uses what's called a Ricker wavelet filter to selectively emphasize features of that specific size. The software then compares the outputs from both methods and reclassifies any overlaps to make sure every aggregate is categorized and counted accurately.
To validate its performance, the pipeline was tested on simulated images that were designed to mimic the properties of the experimental data. In those tests, the algorithm achieved a sensitivity greater than 82% and a precision greater than 94%. It also proved to be more effective than standard Gaussian fitting for estimating local background signals, which is a critical capability when working with low signal-to-noise data like this.
So, now that we've established the methodology, let's turn to the results from its application. The ASAPD platform was used to analyze post-mortem brain tissue from the anterior cingulate cortex of individuals with Parkinson's disease, as well as age-matched healthy controls.
The scale of this analysis was quite substantial. From nearly 12,000 high-resolution images, the platform was able to identify and quantify over 125,000 large aggregates and more than 1.26 million nanoscale assemblies across all the tissue sections from both the Parkinson's and healthy control brains.
Now, consistent with what we already know from neuropathology, the number of large aggregates, those big enough to be resolved as larger than the optical diffraction limit, was substantially higher in patients with Parkinson's. The data show an average of about 26,314 large aggregates in PD samples compared to just 3,866 in healthy controls. That's a nearly seven-fold increase.
But here's where a notable finding emerged. The total count of the nanoscaled assemblies was much more comparable between the two groups. The platform detected about 682,000 of these objects in the PD samples and 582,000 in the healthy controls. This observation suggests that these small assemblies are actually present under both physiological and pathological conditions, which means we need a more detailed analysis of their physical properties.
Now, while the platform can detect objects far smaller than the diffraction limit, it can't resolve their exact size. However, the researchers established a strong linear proportionality between the size and brightness of the larger, resolvable aggregates. So based on that relationship, the integrated brightness of each nanoscale assembly was used as a reliable proxy for its approximate size.
An analysis of these brightness distributions revealed a key difference. Compared to the healthy controls, the distribution of assemblies in the PD samples had a higher median brightness. It was also significantly broader, which you can see from the wider interquartile range. This demonstrates a clear shift toward brighter and therefore presumably larger nanoscale assemblies in the disease state.
To quantify this shift, a disease-specific brightness threshold was defined. This threshold was set at 10,280 photons. Now that specific value corresponds to the 99.7th percentile of the brightness of all assemblies measured in the healthy control samples. So any assembly brighter than this is exceptionally rare in a healthy brain.
Applying this threshold isolated a very distinct subpopulation of bright, nanoscale assemblies. These objects accounted for 9.7% of all the assemblies measured in the Parkinson's disease samples. In stark contrast, they made up only 0.26% of assemblies in the healthy controls. This discovery of a bright, disease-associated subpopulation was a consistent and robust finding across all the samples.
So this quantitative observation of a distinct subpopulation prompted further investigation. They needed to confirm its biochemical identity and figure out its spatial distribution. To do that, the team used a series of complementary biochemical assays and the high-resolution mapping capabilities of the ASAPD platform.
And the results from several orthogonal biochemical tests provided some convergent evidence. First, proximity ligation assays confirmed the higher concentration of aggregated alpha-synuclein in the PD samples. Then, seed amplification assays showed that certain protein fractions from PD brains could seed further aggregation, a key pathological property, while fractions from healthy brains could not. And finally, a portion of the bright aggregates in PD tissue were resistant to enzymatic degradation. This all confirms that these small assemblies in PD are biochemically distinct.
The ASAPD platform also makes it possible to analyze the spatial organization of these aggregates. One way to do this is with a spatial clustering test, which statistically compares the observed distribution of objects to a completely random one. If the likelihood value is greater than 1, that indicates that the objects are clustered together more than you'd expect just by chance.
And this spatial analysis was quite revealing. While nanoscale assemblies in general did show some degree of clustering, that disease-specific population of bright assemblies exhibited a substantially higher degree of clustering. This tells us that these pathological aggregates are not just randomly distributed. Instead, they're organized into specific hotspots within the tissue microenvironment.
So to identify the cellular components of these hotspots, the team co-stained the tissue with markers for specific cell types. The analysis showed an increased probability of finding these disease-specific aggregates localized in close proximity to neurons, microglia, and astrocytes, but conversely, there was a low probability of finding them near the nuclei of oligodendrocytes.
So, what's the significance of all these combined findings? Well, taken together, this evidence lets us construct a more detailed and refined model of the alpha-synuclein aggregation pathway in the human brain. And it offers new clarity on the role of these nanoscale species.
This large-scale dataset confirms that a continuum of aggregate sizes exists within the brain. And critically, it demonstrates that small, nanoscale aggregates are by far the most abundant species. In disease tissue, they actually outnumber large aggregates by more than 16 to 1. This really underscores the importance of figuring out their specific connection to the disease process.
So the data support a model of pathogenesis progression. It seems that small assemblies of phosphorylated alpha-synuclein exist under normal physiological conditions. But in Parkinson's disease, a fraction of these assemblies transitions into a disease-specific state. This state is defined by that increased brightness, seed competence, and spatial clustering near specific cell types. And this subpopulation may well represent a critical intermediate on the pathway to forming the large fibrillar structures found in Lewy bodies. The ability to directly visualize and quantify this disease-specific subpopulation provides a powerful new tool for research. As the authors themselves state, the platform can now be used to design mechanistic studies to understand how these species are created, what cellular microenvironments promote their formation, and how they contribute to the temporal evolution of Lewy body pathology.
But to fully realize this potential, it's necessary to consider the current methodological limitations and the challenges that remain for future work. Acknowledging these factors is just a critical part of the scientific process, and it helps define the next steps for the field.
The key methodological limitations include the fact that aggregate size is inferred from brightness rather than being directly measured. Also, the detection antibody is specific to the serine 129 phosphorylated form of alpha-synuclein. Other forms might have different distributions. Furthermore, visualizing the fine structure of these assemblies will require super-resolution microscopy, and this initial analysis was confined to a single region of the brain.
To expand this work and create a foundational aggregation map of the entire Parkinson's disease brain, a significant increase in scale will be required. The authors estimate that an approximate 2 order of magnitude, or 100-fold increase in the throughput of the ASAPD method will be necessary to explore multiple brain regions across a larger number of cases.
In conclusion, the development of the ASAPD platform enables the direct visualization of a previously hidden, disease-specific subpopulation of nanoscale alpha-synuclein assemblies. This new capability now makes it possible to address one of the central questions in the field. What are the specific cellular microenvironments and molecular pathways that promote the transition from physiological assemblies to the seed-competent, disease-specific species that are hypothesized to drive Parkinson's pathology?